All right, just give me 1 second, guys. I'm just finishing getting set up and then we will get started. Okay, let's see. We left off chapter 2. I realized when I was sending out the emails on Tuesday, I was running late, right? And you guys, I emailed because you're my 9 a.m. on Mondays, and I don't have you until 11:30 on Thursdays. So I had flipped you guys with my other class that I was running late for. Also, I realized that so someone in my other classes told me that like they overheard me yelling at my dog Raven and thought it was my like daughter. Just to be clear, like my dog is Raven, my daughter is Eloise. I was like yelling at Raven because she was trying to eat Eloise's banana. Well, like I was teaching remotely. I was like, Raven, if you eat that banana, I'm gonna hit you. And apparently I came to class and everybody was like, oh, I can't believe you were gonna hit the baby. I was like, what? So just to be clear, my dog is Raven, my daughter is Eloise. Yeah. So I also have a bit of a cold. I apologize. Course projects, assignments. I'm gonna email this out to you guys as well, but this way you guys can like, I don't know if you take a picture or whatever. For the codes, the textbook access codes, I know that I just got new codes, but Remy didn't check to see who had redeemed what. So I have to go through and see. Basically, I have to see who was double assigned codes and then send them out. So if you haven't gotten a code yet, still, I apologize. I got these at like 9 o'clock last night. And like I said, I have a cold and I'm trying to figure it out. But you should be able to do all of the homeworks and everything from the PowerPoints. What else? Our exam got moved. So originally the exams were week 3, day 2, which would have been next Thursday, but we don't go to campus on Thursday. So our exam is moved to not next, like this upcoming Monday, but the Monday after. I think it's the 24th, maybe 22nd, something like that. And so it'll be during class time, which means we get an extra day of review for this exam. Which means also that our next exam, we're going to have a little less time for to do material-wise. So just be prepared for that. But it does mean you get more time to study for this first exam. Okay. I think that's all I have for announcements. So, and I'm going to send out the course project assignments as well to you guys through email, so you will have it in writing. Okay. We left off with chemical reactions, but we skipped to synthesis and decomposition and all that so that we could do that in-person demo thing. Okay. All right, cool. So we're backtracking a little bit. So we're going to chemical reactions. So you should be on page 4 of your packet. So defining metabolism. The metabolism is just the sum of all the chemical reactions in your body. Okay. You can subdivide it into two different categories. So you can subdivide it into what's going to be on the exam chapters 1, 2, and 3. We're gonna also be putting out like a worksheet packet. For the first exam, it's pretty much just your packets, to be fully honest, because there's only 3 chapters on the exam and they're kind of overlap a lot. So we just take the stuff from the, here, I can pull up last semester's 16 handouts. So it looks something like this. And so it'll have like what we're focusing on for the exam. You can just go through, work through it. I would recommend trying to do it without your notes first, and then going back and filling in whatever you don't know afterwards. Okay. I personally don't go through the packets in class because what I find is it defeats the purpose of the packet, because then nobody actually does it on their own. And the whole point of the packets is for you guys to figure out where your knowledge gaps are, right? That doesn't mean I'm not happy to go through the packet with you. Okay. If you have questions about the packet, I'm happy to go through that with tutoring, or if we have extra time after like review or whatever, like we can talk about whatever your questions are, but like specific questions. Like I'm not going to do the packet for you in front of you guys. Oh, which also brings me to another point. So when you guys, like, I'm sure a lot of you guys are like lost right now, right? Which is perfectly fair, right? But it doesn't help me to hear like, I just don't get it. Okay. I'm happy to go through the material with you guys again. That's not a problem. But I need to know like what is confusing me, right? Because repeating my lecture, just like re-saying the lecture the same way that I said it before, isn't going to help you guys. You know what I mean? So if you don't have like a specific question, right, I can't know how to help you guys better. So if you come to tutoring and you're like, I don't know it, you know what I mean? That's fair. But knowing like what part is confusing to you is not only helpful to me, but it's helpful to you. Okay, because it can really help you feel a little less overwhelmed if you can break down specifically where your point of confusion is. If you need to hear the whole lecture again, that's fine too, but we have recordings for that. You know what I mean? And that way you can like rewind, you can play it slower, you can do whatever you need to do. The exam is week 4, day 1 now. Monday. Let me look at my calendar. 9/22. Okay. So yeah, I would just say before you come to tutoring or anything like that, write down any specific questions you have. Or before we have our review session, we're going to have in-class review sessions. Just bring specific questions, because if I know you guys have specific questions, that's stuff that we can like focus on, and like I can help break down a little better, you know? Okay. Anyways, sorry, back to chemistry. So metabolism is going to be the sum of all the chemical reactions in your body. Okay. It is going to consist of 2 things. You have catabolism and you have anabolism. Catabolism is going to be breaking things down into smaller things. So the way that I remember this, cats break things. Catabolism is breaking bonds. When you break these things down, you are releasing energy. Okay, so you are withdrawing from the bank. As opposed to anabolism. Anabolism, you are building things up. Same thing like when you build a wall or something like that, right? You need to use glue or cement or something to stick things together, okay? Same thing when you are building molecules. You are using that energy. That energy is acting like the glue. A lot of the time, you get this energy from the ATP, okay? So the ATP releases that energy and you use it to build things.
We can't see your screen anymore.
You should be able to just see the PowerPoint. If you click on, so like in Engagely, there's like 3 tabs. There's class gallery, there's file share, there's tables. You should be able to click on file share and it should show up for you.
Okay.
Yeah. All right. So chemical reactions. Okay. We talked about these in person already, right? Synthesis reactions. 2 people come together and they're holding hands. Same thing. In order to form that bond, you need to use glue, right? You need something to make those 2 things stick together. Next, you have decomposition. That is when you are breaking the bond. That's when those 2 people step away. They released hands, okay? When you break that bond, you release that energy that was stored in the bond. All right, so you withdraw that energy from the bank. And then exchange reactions, you have 2 people switching spots, right? So you're both making and breaking those bonds, and you are using as well as releasing the energy. Okay. So don't worry about this note with the arrows. You guys don't need to know that. I just forgot to take it off. You have our first MCQ. Just go ahead and click directly on the screen for me. Okay, 10 more seconds. All right, so the correct answer to this one is actually going to be A. All right, so a chemical reaction, we are talking about decomposition, synthesis, exchange. Okay, things were moving around and making new molecules. The other 3 are all going to be examples of bonds, right? So covalent, coparent, right? In covalent bonds, electrons are going to be shared between atoms. Ionic, you're talking about transferring. What do you make when you make an ionic bond? Like, what do the atoms become? So shared is going to be covalent, right? It's in the word ionic.
Ions.
Ions, exactly. They're charged atoms. Okay. So when you move those electrons around, those atoms are going to be charged, and those are going to create ions. Okay. Hydrogen bonds, those are what happens between water. That just basically results in water sticking to itself. Okay. Let's do like 10 more seconds. Also, while you guys are answering this, so first week of classes is over. Like starting today, I have to be more stringent or stringent, whatever the word is for like more strict. on like the clicking into MCQs and being responsive during class and whatnot. So at a bare minimum, please make sure that you are clicking on the MCQs. Don't just like open up the class on your phone and then leave it open and think that's going to give you participation. I have to go in and grade participation, but I've been trying to get the exam ready for you guys and everything and whatnot. So, okay, that's fine. If you miss one, you're fine. It's like if you miss a bunch, that's a problem. It's pretty easy to tell when like somebody's like not clicking on any of them. You know what I mean? But yeah, so the correct answer to this is true, right? 50% or more of an adult's body weight is water. It varies a little bit between men and women. So for men, they have a higher body water weight percentage than women. Women, it's higher in fat. I switched the order of these slides, and I don't like the way that it's switched to. So I'm going to start with this one, and then I'll go back to the last one. For the pH scale, okay, when we talk about acids and bases, a lot of this is coming from the movement of protons, which like you guys don't need to worry about, okay? I just want you guys to understand the like pH scale, essentially, okay? You don't need to know the chemistry behind acids and bases. Now, the pH scale is going to range from 0 to 14. I don't know why it goes from 0 to 14 and not 0 to 20 or 1 to 20 or whatever it might be, okay? This is the numbers that they gave us. So if a solution has a pH of exactly 7, meaning it is like pure water, there is nothing in it to sway it to either side of 7, it is neutral. If it is 7.1, it is not neutral. If it is 6.9, it is not neutral. If it is 6.95, that is also not neutral. Okay? It needs to be exactly 7. If the pH is less than 7, it is called acidic. So for example, hydrochloric acid, that is the strongest acid known to man, is what we measure other acids by. It has a pH of 0. If a solution has a pH of greater than 7, it is called basic, which is also called alkaline. You need to know both of the words. Okay? So sodium hydroxide, that is the most basic solution. That is 14. It has a pH of 14. Okay. Now, it is really important for your body to keep certain areas of the body within a narrow range of pHs. So for example, your stomach acid. Okay, you'll see your stomach acid's all the way at the top here. It goes from 0.9 to 3.0. Your blood, on the other hand, your blood has a pH of 7.4. Okay, what is the mechanism called that keeps things within a narrow range? We talked about it in chapter 1. Starts with an H.
Homeostasis.
Homeostasis, exactly. Is this an example of negative or positive feedback? Negative, exactly, right? There are 2 examples of positive feedback. They are blood clotting and childbirth. Everything else is negative feedback, okay? So your pH of blood, the pH of blood is 7.4, okay? I have a cadmium to help you remember. You're a 10 on the pH scale, maybe because you basic, right? I always tell people, now you can throw chemistry insults whenever you get into your next fight, right? Acids and bases come into play quite a bit in nursing, particularly acidosis and alkalosis. Well, metabolic acidosis and alkalosis. What I mean by that, like I said, you need to keep your pH within that narrow range for the different areas of your body to work, right? If you are throwing up a bunch, right, you lose some of that stomach acid. Okay, when you are throwing off, like you are vomiting off some of that stomach acid, it's going to swing your body pH to the other direction. So you're going to become more basic. That is going to result in metabolic alkalosis. Metabolic acidosis, we even mentioned this before. Diabetic ketoacidosis. Okay. We talked about diabetes when we talked about homeostasis, right? If you get diabetic ketoacidosis, it's not good for you. Okay, so acids and bases come into play quite a bit in nursing. So let's do like 10 more seconds. Yeah, so if something has a pH of 7, it is considered neutral. Okay. If it has a pH of greater than 7, what is it considered?
Or alkaline.
Yeah, basic or alkaline. Exactly. Remember, if you're a 10 on the pH scale, maybe, or wait, you're a 10 on the pH scale, maybe because you're basic, whatever it is, right? I was told that cadmium did help, so I don't know if they're just like, saying, but, well, 10 more seconds. Even if you're not sure, just click on something so you get credit for answering the MCQ. You don't need to type the answers in chat, just click on the screen. Okay, so the pH of blood is 7.3 to 7.5. This is slightly basic, right? Because it is higher than 7. Okay. So that is all for chapter 2. Upload chapter 3. Let's go.
Wait, I'm confused. Could you go over that again?
Yeah, like
You
what part of
said
it's
when the
confusing?
pH is less than 7, it is acidic, right? I thought that's what you said.
Yep. If it's less
Okay.
than 7, it's acidic.
And then what was that question again? That you got basic
So
for that one?
yep, the correct answer was basic for that one. So it's what is the pH of, or so the pH of blood is 7.3 to 7.5.
Okay, so
Or
that's great. Okay,
7.4.
thank you.
Yep, so
Okay,
it's basic.
thank you.
No worries. Okay. So next we're going to talk about cells. I don't remember if I mentioned this to you guys. I realized that the coloring page printed out like blank, but I didn't realize it until I had already printed out 100 copies of this packet. So I will print out the coloring page for you guys, and I'll have that for you to add to your packets later. But for now, I wanted you guys to have the packets for notes. So For the review packets for the exam, no, you guys print those out on your own. And then like the notes packets I'll print out for you guys in class. And then the like guided notes packets, no, you don't need to give those back to me. I don't collect your packets at all. There is like a printer on campus for students, I believe, on the second floor. I don't know if there's like a password or anything like that, but you know that you guys do have a campus printer. So. Yeah, okay, great. So cells are the smallest units of all living matter, right? They're the living things. I put the wrong answer choice for that, so that's all. But this is an Amoeba Sisters video. It's about 10 minutes long. It's really nice. I like the way that they draw things, and like they have nice little animations, and I use Amoeba Sisters quite a bit in this class. Yes, these are all very similar to the questions that are on your test. I'm not going to play it for you guys because we're a little bit behind, but I will send out the links for it. For cell variations, you see these stereotypical pictures of cells drawn, right? But not all cells are drawn, like not all cells are exactly the same, okay? They all serve different functions. In order to serve those different functions, they all look a little bit different depending on what they do, okay? So for example, the nerve cells, okay? The whole purpose of a nerve cell is to transmit information across a long distance. So in order to do this, they have this like long tail thing that comes off. Okay, you don't find this in other types of cells. Red blood cells. Red blood cells are going to be participating in gas exchange and nutrient exchange, but they also have to be really, really, really small in order to fit through like our capillaries. So they have this shape where they have like this inverted donut kind of thing. It's hard to draw with this thing, but this gives them a lot of surface area for that exchange to happen without them having to take up this massive amount of space in order to do it. White blood cells. White blood cells have all of these little dots inside of them, okay? The dots are going to be like, we'll talk about them in a little bit. They're called lysosomes. They help clean up debris, okay? The whole point of white blood cells is to have like an immune function. So by having a bunch of those little dots inside of them, they're well adapted to having immune function. Okay, so pick your favorite example. The whole point is just to explain that cells all look different based on what they do. So function dictates form. You don't need to know these, don't worry about that. This is what the coloring page looks like. It's straight out of your book. So we're going to go into detail about all of these different organelles that I want to highlight for you guys. If you guys want to go back and color it in later on, you can, or just like draw arrows to it. You're more than welcome to. Like I said, I'll print out the page for you. Okay, cells basic structures. So to get us started, we'll do a quick overview of these three. Go back to, you guys have the PowerPoint also. Yeah, I sent it out to you guys on Tuesday, so it should be in your email. Yeah. Sorry, I'm really stuffy. So the cell's basic structures, plasma membrane. The way that I like to describe cells, they're like if you put a Ziploc bag and you fill it up with Jello, okay? The Ziploc bag is going to be the boundary of the cell, aka the plasma membrane. Okay, that's what's going to keep all the stuff inside. Then you have your nucleus. Your nucleus is going to be like the center of the cell. So it's like if you drop like a, I don't know, like a jawbreaker or lemon head or something in that bag of Jello, that is going to be your nucleus. The cytoplasm is going to be a gel-like substance surrounding the nucleus. This is going to be the Jello. Okay, so this is what everything is floating in. This is where all your organelles and molecules are, each of which have their own job to do. Now, besides defining the boundaries of the cell, okay, the plasma membrane is also going to be responsible for regulating the passage of substances into and out of the cell. All right. It doesn't do this by like just putting up a wall. Okay, so when we say like, should the plasma membrane be thought of as a fence or a wall, it should be thought of as a fence because it has this characteristic called selective permeability. Okay. Selective permeability just means it gets to pick and choose what comes in and what doesn't. So some things are allowed to come through really easily. Some things are not. Some things it keeps blocked out. All right. So the reason why we think of it as a fence as opposed to a wall, fences have like little holes in them. They have like these little like spaces in between those slats, right? Where things can pass through. Your wall should not have holes in it. All right. If it has holes in it, that's just bad construction. Or you need to get that wall replaced. I don't know. So for defining the boundary of the cell, the plasma membrane also regulates the passages of substances into and out of the cell. Okay. Define selective permeability. Selective permeability means that some substances can pass through easily while others do not. And it should be thought of as a fence, not a wall, because there's like spaces for things to go through. All right. I typed those things out in the chat. So you can kind of think of as your plasma membrane as your border patrol. All right. So they're the things that are monitoring what comes in and goes out of the cell. It is primarily made of what are called phospholipids, but like they're just a subcategory of what are called lipids. All right. Lipids are just another word for fats. We used to talk about like lipids and carbohydrates and all that stuff in chapter 2, but we don't come across them again really until we get to chapter 22. So we decided to like take it out of chapter 2. So if your book starts talking about like lipids and carbohydrates and whatnot, like you're supposed to know what they are, that's why. Okay. You can also find things like proteins. Okay. They can act like tunnels, act as receptors, so they receive information. They can act like signals for other cells. And you can also find cholesterol, important for membrane fluidity, membrane structure. Okay. The point of this question, membranes are not just one thing. There's a bunch of stuff in the plasma membrane that lets it decide what is allowed to come in and what is not allowed to come in. Okay. That's the whole point of it. All right. Now, the plasma membrane is really important when we talk about nursing because the plasma membrane is really going to help you prevent infection by acting like that physical barrier, right? Just having that bacteria or that virus next to your cell isn't necessarily going to mean that the cell is infected. What it means is that like the infection comes when those bacteria or those viruses or whatever get inside of the cell. So when they cross that plasma membrane, if they have not crossed that plasma membrane yet and have not gotten into the cell, it's much better off, you know? So keeping that plasma membrane intact is really important for disease prevention. All right. A lot of viruses and bacteria have certain like adaptations to like let them break through the plasma membrane so that they can get inside the cell. Okay. Next thing we're going to talk about is the nucleus. The nucleus is the cell's control center. Okay. That's going to be that, like, I don't know. I told you, like, we dropped a jawbreaker in our bag of Jell-O. It'll be the jawbreaker. Okay. I made like a diorama of a cell when I was in like high school or something and a candy. So I talk about candy a lot during this lecture because it's always stuck for me. So the nucleus is the cell's control center. It is going to contain all of the cell's genetic information, aka DNA. All right. So I'm sure you guys have heard of DNA testing or like the 23andMe kits and whatnot. This is what it's testing. It's the genetic information inside of your nucleus. Okay. Most cells are only going to have one of these guys. Sometimes they have multiple nuclei. And we'll explain those when we get to them. So, nucleus is the cell's control center and contains all of the cell's genetic information, aka DNA. Just as a fun fact, mature red blood cells do not have a nucleus. They lose it during maturation. I'll give you guys a minute to catch up on writing as well as answer this. Okay. So 10 more seconds. Yeah, great job. So, a cell's genetic information is contained in the nucleus. Okay. Now, there's a bunch of different organelles, so meaning little organs, inside of your cell. You guys do not need to memorize all of them, okay? We just want you guys to know these 5. So we narrowed it down to a pretty small list, okay? So starting with your endoplasmic reticulum. Endoplasmic reticulum has 2 types, which are defined based on their appearance, which is thanks to these things called ribosomes. Okay? Ribosomes are going to synthesize protein. And they are found in the cytoplasm, like freely floating. And they're attached to rough endoplasmic reticulum. Okay? So ribosomes are going to synthesize protein, and they are either found in the cytoplasm, like freely floating around, or they are attached to the rough endoplasmic reticulum. Okay? So rough ER, therefore, has ribosomes. Since we know now that ribosomes are making protein, it makes sense that rough ER is also going to be synthesizing protein. Okay? So rough ER has ribosomes. It is also synthesizing protein. Smooth ER, on the other hand, does not have ribosomes. and it is responsible for synthesizing lipids, aka fat, and carbohydrates, and does some detoxification.
Okay, wait, because I'm getting confused. I feel like you just talking to me and I don't know what to write. So you telling us that the ER have a rough ER and a smooth ER, correct? ER, correct?
Yep.
Okay.
So the way that I like to think about them is like the two types of candy. I told you I talk about candy a lot. So you know how like there's like that dot candy from, I don't know if they still make it. I assume they do, where you have like the little dots that you pull off of the ribbon. Like I'm gonna assume everybody's like nodding their head. Okay, that's like what the rough ER is in my mind. Okay, you have these like ribosomes stuck to them. Okay, smooth ER on the other hand is just like the ribbon candy that doesn't have the dots on it. It's just smooth. Okay, so it's named based on what it looks like. They both serve two somewhat different functions. They make things, but they make different things. And I typed the way that I would summarize these things in chat for you guys as well to help like with notes. So okay. Okay, so next we have our Golgi apparatus. Golgi apparatuses are going to receive proteins from the ER and prepare and package them for export to other parts of the body. Okay. The easiest way I like to explain this, it's like the Amazon warehouse. All right. So Golgi apparatus, the easiest way to explain what it looks like a stack of pancakes. Or a stack of whoopee cushions that have been like squished together, stuff like that. Okay. It is going to be responsible for repackaging, sorting, and repackaging proteins. Okay. So the way that I explain this, right? You go on Amazon. I love Amazon. I order a bunch of different things on Amazon. Okay. So let's say I order a candle, a pair of scissors, and a hat. Okay. The same manufacturer is not going to make all three things, right? So those manufacturers are going to send those things to the Amazon warehouse. All right. So the manufacturers are going to send the candle, the scissors, and the hat to the warehouse. My order is going to come into the warehouse, and the warehouse is going to pull out the things that I've ordered. Okay. And it is going to stick all three of them together in a package, and it is going to stick a label on that and ship it off. All right. These packages are called vesicles. So you can just write that in there as well. Packages. Yeah, you know what? Just leave it. It's fine. We'll see vesicles later on too when they come up. But
So if you was to ask us this on the test, what would you say? Like, because I'm supposed to just write down a stack of pancakes on my notes.
it's just like what they look like. Mainly, we would probably ask you like what the function is, like which organelle is responsible for sorting and repackaging proteins.
Oh, okay, gotcha.
Yeah, the stack of pancakes is just so that it like sticks in your head better.
So a question would be like, what something like what you just say? What
Like
package?
which organelle is responsible for sorting and repackaging proteins?
Okay, wait, let me write that down.
Okay. So the whole point is the Golgi apparatus is like the Amazon warehouse. You want to write down Amazon warehouse, you can. But just know that it sorts and repackages proteins. Ten more seconds. Yeah, so smooth endoplasmic reticulum, right? Smooth is going to have that smooth appearance, meaning it does not have the little bumps. The bumps are showing up because of the ribosomes. Okay, next we have centrioles and lysosomes. Centrioles are going to be bundles of microtubules that play a role in cell division. Okay. Do not worry about anything about them beyond that, truly. Okay, just know that they play a role in cell division, and that's it. Okay. You guys don't need to know what they look like, mechanics of them, anything like that. Lysosomes. Lysosomes are also often called cellular garbage disposals. Okay. They contain enzymes. Enzymes to help break down protein the cell doesn't need. Okay. So if you remember when we talked about like form dictates function and we talked about white blood cells and how they have all these little dots inside them to help clean stuff up, this in part is due to the lysosomes. Okay. So one way you can remember it, lysosomes sound like Lysol. Lysol cleans. Lysosomes are responsible for cleaning. Okay. Next, we have our mitochondria. Mitochondria are often called the powerhouse of the cell. Because they are responsible for producing ATP. Producing ATP. ATP stores energy, right? We talked about it in chapter 2, like right at the end of last class. Probably as people were getting sick of hearing me talk, but yeah, ATP is going to be responsible for storing energy. Okay. Next, we have our cytoskeleton. Okay. The cytoskeleton is going to be responsible for, or it's the supporting framework of the cell. Okay. Skeleton, supporting framework of the cell. All right. It's going to be responsible for a couple of different things. It determines the cell shape. It strengthens the cell. It allows the cell to move in some cases. And it can have a couple different extensions called microvilli, cilia, or flagella, which we're going to talk about. And I realize that these pictures are blank as well. So you can either like, yeah, I would just like draw them in. They're not like super complicated. So again, not that you're going to be asked to identify like which one of these are microvilli versus flagella or whatever. Just might help you understand better like what the function is. Okay. Now, first you have microvilli. Okay. Microvilli are just little like hills that stick up that are really, really, really small. The whole purpose of microvilli is to increase the surface area of the cell. Okay. You can find them in areas of your body that's responsible for absorbing like nutrients. So in your intestines. Cilia, on the other hand, cilia are going to move, but they do this like crowd surfing function. Okay. So let me see. So cilia move particles along in a crowd surfing motion. Respiratory tract will move mucus. Okay, so cilia is going to move particles along in a crowd surfing motion. For example, the respiratory tract, where it's going to move mucus. Okay. Lastly, you have your flagella. Flagella cause the cell itself to move. Only find them in human sperm. Okay. That is the only place you will find them in the human body. All right, give you guys another minute to finish writing everything down.
Will these be on the exam?
Yes.
Okay.
Yeah, so the ATP is going to be generated in the mitochondria, right? Mitochondria is the powerhouse of the cell. You've ever seen those like memes? Or maybe not. I think I'm crazy right now. It's fine. Ten more seconds. All right, yeah. So cellular garbage disposals, lysosomes, right? Lysol, lysosome. Okay, 10 more seconds. Yeah. So flagella is considered an extension of the cell.
Okay, so what would cytoskeleton be? Because that was on the other slide, right?
So a
I
flagella
didn't
is
write
part
that
of the
down.
cytoskeleton. So let me go back. So here we go. So
Okay, that's the same
cytoskeleton,
word.
yeah. So a flagella is a type of extension from the cytoskeleton.
Okay, wait, let me write. You said the type of. Okay, I got that already. Thanks.
Yeah. So it's like you have like an apartment and then you have a balcony, right? Like the balcony is still part of the building, but it's still like its own thing. So the flagella is kind of like the balcony in this case.
And I see that you put those bullet points in the chat. Do we need to know those? Like the strengthens the cell, allows cells to move? Should we write
I
that
mean, I
down?
have them. Yeah, I mean, I have them in your packet. That's why I was typing it in the chat for you guys.
Okay, because I'm writing in my notebook.
Yeah.
All right.
Excuse me. Okay. I think now is a good break point before we get into transport mechanisms. because transport mechanisms take a minute to get through. So let's go ahead and do our 10-minute break now. I will put a timer up on the screen. And I will see you guys in 10 minutes. Okay? Okay, let's go ahead and get back into it. Okay, so next we're going to talk about how things move across the cell membrane. Okay, so these are called transport. You can have two types. You have active transport and passive transport. Passive transport is exactly what it sounds like. Passive. I spelled passive. Hold on. It is passive. It does not require energy. This can include diffusion, osmosis, filtration, and facilitated diffusion. Okay. We are going to focus on osmosis more so than anything else. I have the other ones just fully typed out for you guys so that they're like there if you want to know more about them. But we really want you guys to focus on understanding osmosis. So we took out a lot of the other stuff. It's still in your packets because like we'll still like briefly go over them, but focus on knowing osmosis over all of them. Active transport. When you are active, you require energy, right? You have a lot of energy. That is what this is. Active transport requires energy. Energy. It includes transport by pumps and vesicles. Okay. So passive transport does not require energy. Active transport requires energy. Type those out in chat for you guys as well. Okay, like 10 more seconds. Yeah, so active transport requires energy, right? Passive transport does not require energy. We are going to focus on osmosis, all right? Osmosis involves the movement of water from an area of low concentration to an area of high concentration. Involves the movement of water, an area of low. Now, what I mean by this, okay? This is different than diffusion because diffusion, you can think of diffusion as like a dye tablet, okay? If you put a dye tablet in the water, right, it's going to diffuse out, meaning it's going to spread out on its own, all right? Osmosis, on the other hand, happens when those particles, so everything wants to be equal, right? But those particles can't move to a certain extent. So instead, what happens is the water moves to equal it out, okay? Because you want everything to still be balanced. But if something can't pass through the membrane, for example, our plasma membrane, right? Certain things can't pass through the plasma membrane. So the water is going to move to equal that out, okay? So, for example, albumin, it's a protein that It gets produced by the liver. You guys don't really need to know albumin, but okay, that's fine. Basically what happens is you just want to look at everything. So if you have like this just specifically says albumin, but if you also have salt on that side of the membrane or whatever else, it'll take that into account. Okay. The water is going to move to balance out the two sides. Okay, so trying to think of I feel like I'm just repeating myself over and over. I have a really good Amoeba Sisters video for you guys, so I'll play that for you in a minute. But basically, you hear osmolarity referred to in terms of what's called tonicity. Okay, tonicity is how a solution affects the movement of water across a membrane. So, even though these solutes cannot cross the membrane, they are still going to impact how the water moves. Okay, so tonicity. How? What's going on? Sorry, I'm trying to pull up the osmosis. I thought I had the link like already. Why is it doing this to me? Sure. You guys don't need to know the pressure and all that. Sorry, I meant to stop it before then. Yeah. So once it gets to like this part where it gets to pressure, you're good. Okay. So no. So basically, osmolarity, the whole point of it is water is moving. Okay, so movement of water, you are talking about osmolarity, okay, or osmosis. Now, in nursing, it is really important because this comes into play when we talk about IVs, like they said in the video, okay? If you have high osmolarity, it would indicate dehydration, okay? You don't have enough water in your bloodstream, okay? If you have low osmolarity, it means that you have too much water, not enough particles, okay? So this comes into play when we talk about IVs quite a bit. Isotonic solutions. Iso means the same. Okay, so this is when the concentration of solutes is the same outside of the cell as it is inside of the cell. Okay, so isotonic solutions, it's not that the water's not going anywhere. All right, it's that there's an equal amount of water moving in as it is out because you already have a balanced amount of solutes. Okay, so ISO, same. Same inside as it is outside. Hyper. So hypertonic. You have a higher amount of concentration of solids. Sorry, you have a higher concentration of solids outside of the cell as compared to inside of the cell. All right, so in order to correct this, the water is going to move out of the cell to balance it out. This is why you don't give like super, super salty water in your IVs because it is going to pull that fluid out of the red blood cells and cause them to shrivel. All right. Hypotonic. Hypotonic is going to contain a lower concentration of solutes compared to the fluid inside of the cell. All right. This is why you don't give pure water for what is it? You don't give pure water for IV solutions. Okay. So let me think of a way to ask this. If something has a high concentration of solutes, meaning it is hypertonic, okay, does that happen when a patient is dehydrated or overhydrated?
Overhydrated.
Overhydrated.
Dehydrated, right? Hypertonic means that you have a high concentration of solutes. So like if you think about like when you make Kool-Aid, right? Because you can see it, right? If you put like a lot of the Kool-Aid solution into like a very small amount of water, it is super, super dark colored, right? As opposed to if you put a ton of water with just a little bit of Kool-Aid, that is low, okay? You are looking at the amount of water and the concentration of solutes. So hypertonic means you have a lot of solutes. So hypertonic means you have little water, meaning that it is very very concentrated. Does that? I feel like I'm making this more confusing. Let me think. Tonic, high concentration, solutes, low concentration. Okay, so hypertonic high concentration of solutes, low concentration of water. That is the like you've loaded up your Kool-Aid mix into like a very small amount of water. Okay. Hypotonic will happen, hypertonic, sorry, will happen when you're dehydrated because you have a low amount of water. Hypotonic, low concentration
I have a question.
Yep.
So basically, when we use it and when we see the word hypo with anything, it's always going to mean low, right?
Yep.
Okay.
Yeah, hypo always means low, hyper means high. So like hyperactive, they have high energy, hyperglycemia, high blood sugar. Yeah.
Okay, cool. Because I was looking at my notes from my other class.
Yep. So hypertonic. Okay. Hypertonic, you have a high concentration of solutes. If you have a high concentration of solutes, you have a low concentration of water. Okay, because we are talking about things in proportion, essentially. If you have a lot of solutes, comparatively, you have less water. All right. So when you put red blood cells in a hypertonic solution, where there's a bunch of solutes and not a lot of water, the water that is inside of your cells is going to want to move out of your cells to try and balance that out. Now, realistically speaking, it's never going to be able to do that because there's not enough fluid inside of your red blood cells to make that balance work, right? So all that ends up happening is your cells just end up shriveling and dying. So you don't want to put it on a high thing. For hypo, it's the opposite. Okay? There's not enough solutes to balance out what is inside of the cell. So instead, water moves into the cell to try and balance it out and keeps moving in until it causes the cell to burst. Okay. And we can spend some time on this during like class in person on Monday. Because I feel like it'll be easier to explain in person.
I want to ask you a question.
Yeah.
Do you think that you probably can what's gonna be on our test, not all of them, but you think you can make some of the questions in like a Quizlet flashcard, like so we can study it that way? Because that like helped me study better, and I think most of the people in our class probably can get it that way too a little better.
Yeah, so the way that I do my reviews is I have multiple choice questions that we go through together, and I put them together into like a worksheet, and we'll go through all of the answer choices. So when
And
we
that would be
go
the
through
questions that's gonna be on
questions,
your test you give us too.
not the exact questions, but pretty similar.
Right. Just worded different.
Yeah.
Cause girl, your class hard. Okay.
Yeah. But no, we do a lot of review. I try to always allocate like 2 days for review for you guys, so that we can go through everything. But you'll see, the reviews are usually pretty like. I feel like people like my reviews. I don't
I'm
know.
following though. I had printed the whole book, the whole chapter off that you sent us when I was at work. So I got it right here too. I'm just going through it, which one, making my notes. But I think that'll be a good help though if you could do it that way too.
Yeah. How do we sign up for tutoring? Because this whole science thing, I'm not catching on to none of this.
Yeah, so if you guys want tutoring, hold on, my bright space logged me out. Hold on.
So what's the difference from you teaching us now than the tutoring?
So the tutoring is like one-on-one. So like I said, if you come to tutoring, My thing with tutoring is like come with specific questions that we can go through together. Because like going through the lecture again to me, like you can just do that in the recording. You know what I mean? You can rewind, you can pause it, you can go at your own pace. For me, I think that it's more helpful when you come to tutoring to like have this is what is confusing me. This isn't sticking in my head because of this, or something like that, right? So when you're in,
Everything is
you're
confusing me.
talking. Right, but if you just tell me everything's confusing you, I don't really have a direction to help you in. You know what I mean? Like I'm just going to explain things the same way I did. And me redoing the lecture isn't going to help you. Because you could just watch the recording if you want to rewatch the whole lecture. But if you can tell me, like specifically, I'm getting confused on bonding because I don't understand what you mean when you say electrons, right? Then we could go back and I could explain to you what electrons are. Stuff like that. So going through your notes and having those questions ahead of time is going to make your tutoring session a lot more effective for you. Let me view this as a student. So if you go to your Brightspace and you go to content, okay, instructor office hours links, you have Dr. A, who's our course tutor, and then you have me as well, okay? So if you click on mine, I do have this set up now. You can click on these 30-minute meetings, tutoring, and it should show you all of my available appointments. So I have some sessions still tomorrow. I think I have people scheduled all Wednesday pretty much. But yeah, you can click through, you can see what appointments work best for you. I think Dr. A has her set up in like hour blocks. Let me pull it up. So A&P tutoring. Yeah, hers are set up in like 1 hour blocks. She has like a lot more appointments available.
So her sessions is the hour and yours 30 minutes.
Yeah, because I have less office hours.
Oh, okay.
Thank you. Yeah. So I try to fit more people into my office hours.
Does she teach at the school?
So she teaches remotely. She's down in Houston, I believe. She's somewhere in Texas. I know that.
How's she
She's
gonna help
taught.
me out there?
Well, she'll just be with you online. She's really wonderful, I promise. Doctor
Okay,
is
I'm
great.
trying.
Okay. And you can always set up appointments with me still, I promise. It's just that I have them set up in 30-minute blocks. Honestly,
It's only
I
30 minutes.
only have like 1 or 2 sessions that have lasted a full hour, so that's why I usually keep them in 30 minutes. Because
Oh, okay.
yeah, but I mean, really, like it's your time. So if you want to set up 2 blocks in a row, you can. I
Oh, okay,
don't
I understand.
once or
That's
twice.
better.
Yeah.
Thank you.
Yeah, no worries. Okay. Some more drawings to help you guys if you like the
I'm
drawings.
sorry, I got one more question. So if I set up tutoring with you, I can do in person with you, right?
Mm-mm.
Oh, still
I only
online?
do online. Yeah.
Oh, so all tutoring is online.
Yeah, because my office hours aren't in person. The only day that I'm in person is on Mondays. And I'm teaching Monday from 9 a.m. to 9 p.m. I don't have a break, so I don't
Oh,
have time to
okay.
do.
Okay.
Yeah.
Thank you.
Yeah, no worries. I knew I had the video in here somewhere. Like I said, we're not going to focus too much on like the other transport mechanisms, like the filtration, diffusion, but just to talk about what they are briefly. Filtration is like when you make coffee, like the coffee gets dissolved in the water and then the water gets pushed through the coffee filter. That's basically filtration. It's the same thing. Solutes get dissolved in your water and then the water gets pushed through the capillary walls. I don't think I even have you guys write anything down for it. Yeah. Don't worry about this question. Facilitate diffusion. Sometimes things just need a little help. And so sometimes we have specialized proteins to help things go across. We were still not using energy with facilitated diffusion. It's just helped out, essentially. Active transport. So with active transport mechanisms, as we said, they use energy because you're not like going down a concentration gradient, meaning you're not going from like, I'm trying to think of how to put this. Like you want to move in a certain direction, right? Like think of how gravity works, right? You drop a ball, that ball wants to go down because it's being pulled in a direction, right? Active transport is when it's going the opposite of that. It's going swimming upstream. It's going against what it wants to do. Okay, so in order for it to do that, it needs to use energy. Okay, so in active transport mechanisms, solutes are going to move up the concentration gradient. Okay, let me type that up for you guys in the chat. An active transport mechanism. Solutes move up the concentration gradient from areas of lesser to greater concentration. Okay. Active transport requires energy in the form of ATP. All right. So we have two types of transport or active transport. Okay. First, we're going to have transport by pumps. This is like the most important example that you're going to see of this is the sodium potassium pump. All right. We see this quite a bit in like neuroscience and whatnot, but you guys don't really like need to know the specific mechanics of it. Okay. Really, what I want you guys to know is that sodium is going to move out, potassium moves in, and it uses ATP to make this happen. Okay. So that's going to be your next like little fill in the blank thingy. All right. So sodium potassium pump. It moves sodium out, potassium in, and uses ATP to make this exchange. All right. Don't stress too much about knowing like the mechanics of the pumps or like pumps in general, honestly. When we need them, we will get to them. Okay. The most important thing to focus on is osmosis for this particular exam, because osmosis is really important when it comes to like IVs and whatnot. This stuff will be more important later when we get to like neuroscience and whatnot. But right now we're trying to like just do the very basic stuff for nursing. Okay. So then you're going to have transport by vesicles. We have talked about vesicles once before already today. Okay. When we talked about the Golgi apparatus, we talked about vesicles. Vesicles are like those little packages. All right. So let's see, where's the fill in the blank? Cells also have the ability to move large particles or numerous molecules through the plasma membrane. This process also requires the membrane. The cell membrane creates a vesicle to transport matter either into or out of the cell. Okay. So your next fill in the blank for that, like active transport, is the membrane creates a vesicle to transport matter into or out of the cell. Ah, why am I gonna go all weird on this? Okay, there are two different types of transport by vesicles. Exo and endo. The words look really complicated, okay? So just look at the prefix. Exo, think like exoskeleton, like in bugs, okay? Exo is going out of the cell. Exoskeletons and bugs, they have skeletons on the outside of their body, okay? So exocytosis is going to release substances out of the cell. So exocytosis okay. Exocytosis releases substances out of the cell. So for example, when mammary glands secrete milk. Mammary glands secrete milk. Endocytosis. Endocytosis is when substances come into. Endo means in. Okay? Bring substances into the cell. All right. So what happens is the plasma membrane is going to trap something that's too big to just pass through the plasma membrane and bring it into the cell. Okay, you have two forms of endocytosis, including phagocytosis and pinocytosis. Do not worry about knowing phagocytosis and pinocytosis. Okay? You guys will never need to differentiate between the two. I'm just going to copy this, put it in chat. So, plasma membrane takes something that's too large to just pass through the plasma membrane, and it pinches off a little vesicle and brings it inside. So those are your transport mechanisms. Okay. Yeah, so your sodium potassium pump is going to be a type of active transport. Okay? So active transport. So meaning that it uses energy, uses that ATP. Okay? Okay, 10 more seconds. Yeah, so osmosis involves the movement of water. Okay, so it's going to involve the movement of water across that plasma membrane. Okay, cell growth and reproduction. So I have a nice little summary table for you guys. It is all typed out here at the end. So just listen for a minute and then we'll get to the thingy at the end. Okay. So as we already said, your cell's genetic information is also called the cell's DNA. Okay, so the DNA is going to store the genetic information. It's okay. If you miss a question, you're fine. So the cell's DNA is going to store the genetic information. Okay. Yeah. DNA is going to be double-stranded, meaning that it has two of these strands. You know what? I'm just going to teach based on the table because I feel like the table shows everything in a nice, like, really summary little, like, put together. Okay. So the DNA is going to store genetic information. Okay. RNA, on the other hand, the whole purpose of RNA is to transmit and copy that information. So the way that I like to explain the difference between the two, it's like if you go to the library, okay, and you're learning to build a wall or a bridge or something, whatever it may be. Okay, you go to the library, you find a textbook on how to build whatever it is that you're trying to build. Okay. That textbook, though, is giganormous. You don't want to take that home. It is huge. So instead, what you need to do is take the pages that you need to copy. All right. You don't want to take the whole textbook home, right? So you end up just taking it to the printer, making a copy of the pages that you need, and taking those home to build whatever it is you need, right? And think of the nucleus as like the library, all right? Inside of the library is the textbook. The textbook is the DNA. All right. So you make a copy of the DNA, just the pages that you need from it, and that is your RNA. The RNA is what you take out of the library into the cell to make all of the stuff. Okay, so DNA is going to store the genetic information, whereas RNA is going to transmit and copy the genetic information. DNA is going to be double-stranded, whereas RNA will be single-stranded. Okay, so D double, RNA is going to be single. For composition, aka which sugar does it have, you don't need to type out the whole sugar, right? Or write out the whole sugar. You just don't. I'm never gonna like give you 4 different D sugars to differentiate between. Just know that the DNA has a D sugar, RNA has an R sugar. Okay, so deoxyribose goes with DNA, ribose goes with RNA. For bases, so both of them, so the way that DNA and RNA work, they have these bases that link up together. And there's like a DNA model in the classroom that I'll show you guys. But they have these bases that link up together. Okay. 3 of them are the same between DNA and RNA. So both of them have adenine, guanine, and cytosine. The last base is different. So like I told you guys when we were talking about the systems, right? You want to focus on what's different between things when you're studying. Okay. So DNA has thymine. So THYMINE. RNA has uracil. URACIL. Okay. As far as the location goes, DNA is going to be found in your cell's nucleus. Don't worry about knowing the mitochondria. Okay, just know that it's in the nucleus. RNA is going to be found in the cytoplasm. Right? So you've taken it out of the library and you've made that copy. Okay. So, again, this is in the PowerPoint that I sent you guys. This is all you need to know about DNA and RNA. All right. Don't worry too much about knowing which bases batch up together. Or anything like that. Okay. If it's not in the table, don't worry about it. Okay. All right. So next we have protein synthesis. Okay. Protein synthesis is going to be divided into two main phases, transcription and you have translation. Okay. Starting with transcription, you go from DNA to RNA. All right. mRNA is just a type of RNA. Translation is going to go from RNA to protein. All right. Do not worry about knowing, like, in detail anything about either of these steps. Okay. All I want you guys to be able to do is identify that protein synthesis is made up of transcription and translation. Okay. So step 1, transcription. Step 2, translation. Those two steps together are protein synthesis. Don't worry about knowing it more in depth. The book goes into a lot more detail than you need to know. Okay. Last thing we're going to talk about. See, this is the diagram from your book. You do not need to know all of this. All right? All you need to know, transcription and translation. That's it. Do not worry about the different types of RNA or what would like copy. None of that. No. Okay.
Oh, I must have missed this, because what's this?
What are the bases? So we said that DNA and RNA have 4 bases, 3 of which are the same. DNA has, I'll just type it out in the chat. DNA has thymine as its last base, and RNA has uracil. All right, like 10 more seconds. Okay. So uracil is going to be found in RNA. Okay. So that's from like this thing here. Okay. All the slides that are before this are the same thing, just typed out like more in depth. But for the purposes of like your table, you can just base it on this slide and you're good. Okay. All right, 10 more seconds. So, like I said, transcription and translation, two main phases of protein synthesis. Okay? Protein synthesis. Now, the last thing we're going to talk about is the cell cycle. But really, we're not going to talk about the whole cell cycle. And I don't want you guys to know the whole cell cycle. The only thing I want you to know from this is mitosis. Okay? Mitosis is just when one cell splits into two. Mitosis is just the process of cell division. Okay? So I'll type it out in chat. Mitosis, when one cell into two. Okay? There are 4 phases of mitosis. All right? We don't do the reproductive system or like, yeah. So we're not ever going to talk about meiosis, which is a different type of cell division thing. Okay? So really all you guys need to know is like the names of these phases and you're good. Okay? So prophase is when the chromatin are getting ready. Okay, so prophase is when everything is doing its prep for cell division. So it's just getting organized. I don't want to like go into all the detail because I don't think you guys really need to know all that. I know that you're probably overwhelmed. So prophase is just when everything is getting ready. So that's that first step. Okay. Metaphase is when everything lines up in the middle. So metaphase, middle. All right. That's how I remember it. Metaphase is middle. Anaphase. Everything starts getting pulled away from each other. So anaphase away. And then telophase is just the last phase. Okay. So telophase, everything starts to reform and the chromosomes. So like, you know how people say like they get chromosome testing or like in Down syndrome there's an extra chromosome. What they're really talking about is these like condensed forms of DNA. Okay, so those are the structures of DNA. We don't really talk about it too much in this class, so don't worry too much about it. Okay, just know that there's 4 phases, and this is their names, and you'll be fine. Okay. So yeah, you have 4 phases in mitosis. Okay. Prophase, metaphase, anaphase, telophase. Okay. So that's all I have for you guys today. We're getting done a little early. Like I said, we're gonna start out with review next week. We'll do review both Monday and Thursday next week. So come with any questions that you guys have. We'll do like a multiple choice review. I always start with crosswords. If you're repeating with me, a lot of people I feel like crosswords either hit or miss, but the whole point of the crosswords is to get you guys to like go through your notes and everything. You'll see. So
I got a question. So I was looking on TikTok over some things. So basically we have this class 2 semesters or just
no,
one? Okay, one. So
just one.
this AP and this AP one.
So a lot of nursing schools, like for RN programs, will have like A and P 1 and 2. For
But we
LPN, it's just in one.
so overall I put AP in one. So basically I was looking on that. They said a lot of people say you can learn from just by writing everything out, the pictures. So basically, writing the pictures and connecting everything together. So, so we learn everything overall for A and P, like the whole system of our body.
Yes, but like we don't go into a lot of detail in this class, if that makes sense. So like it's accelerated, right? Like that's why it's all in one semester. So we don't have like the time to go as in depth in an LPN program that we will in the RN program.
But we need to know that stuff, though, right?
No,
Oh,
not for this class. Yeah, no.
so
If it's not in this class, you don't
so
you
could
don't
you
have to like sorry.
could you just give me a main overview of basically like what's gonna be on the test, what we need to know that's gonna be on the test, like for the end of semester, so we'll know, so I know the main things I have to study on.
Like the learning
Review next
objectives?
week, she said.
Huh?
We doing review next week, she said.
Okay.
Yeah, and like if you ever like really want like a summary, like just look at the learning objectives. They're at the front of your packets. We also have like all of the learning objectives typed out on Brightspace. I think I have to like update the list because we took a lot of them off this semester. But
Also,
like,
basically the learning objectives, what you gave us is basically the stuff we need to learn for the test.
yep,
Okay, okay,
not
thank
the
you.
one in FA Davis because FA Davis has more learning objectives than you guys need. Okay, we
No,
took a
but the
lot of
one,
those out of FA Davis.
so only the stuff, pay attention to only the package you gave us.
Yep,
Okay, thank you.
no worries. All right, guys, have a great rest of your day. I will see you on Monday. Bye,
Okay,
everyone.
bye.